Okay, today we are going to do dihybrid crosses and polygenic inheritance. Both of these concepts are definitely on your mastering this weekend, so that's why I want to make sure that we get it before the week is out. So in order to understand a dihybrid cross, we need to first rehash what a monohybrid cross is. A monohybrid cross is what you've been doing with your traditional 4-grid Punnett Square. It is a cross between various forms of one trait. And I need to be sure you guys understand the difference between forms and traits. In pea plants, there's a trait called height. You can either be tall or you can be short. So how many forms of that trait are there? That's it. The forms are the different ways it can be presented, but the trait is the trait. It's the characteristic. So in this room, I see brown hair, I see black hair, I see blonde hair. Those are 3 forms of one trait, hair color. So don't get trait and form confused. Mono is the number of traits. You can have 20 different hair colors. That's fine. But the key is you're assessing hair color. So if I were to talk about pea plants, a dominant trait is tall, a recessive trait is short. So if I had a heterozygous parent crossed with a homozygous recessive parent, I would ask you guys to make me a Punnett Square to show all the possible offspring. Now before you do that, I want you to take a look at this heterozygous parent. How many different, let's say that's the male. How many different types of sperm cells can it make? What do I mean by that? How many different forms of the height gene can it make? 2. It can have a sperm cell that has the dominant allele, and it can have the sperm cell that can have the recessive allele. And as for the female, the egg cells, it's going to be both recessive. So there's a sperm, there's an egg, there's an egg. So those are easy enough. What you need to get used to now, and this will get you ready for Monday, is you need to identify the expected frequencies of genotype and phenotype. Frequencies means percentages. Remember that there's also a difference between expected and observed. So when it comes to expected genotype, Mitchell, what % of these will be homozygous dominant? 0. Natalie, what % will be heterozygous? 0.5. And Samantha, what % will be, not Samantha, Tanae, what % will be homozygous recessive? 0.5, okay? That's genotype. Now let's do phenotype. Elizabeth, what % are going to be tall? Or would you expect to be tall? 0.5. And Mary, what % would you expect to be short? Right. You guys are going to need to get used to just recording the observed. Now, is that a guarantee? Just because you flip a coin twice doesn't mean you get heads once and tails once. Okay. So that's just the monohybrid cross. That's easy stuff. That's kid stuff. Let's get to the dihybrid crosses. A dihybrid cross is a cross of various forms of 2 traits at the same time. So let's continue with this whole pea plant. The two forms of the two traits we're doing is going to be height. You could either be tall, which is dominant, or you could be short, which is recessive. And we're also going to do color. You could be dominant, which is green. You could be recessive, which is yellow. Now I'm going to teach you the hard way first, then I'm going to teach you the shortcut. You need to know how to do both. It's kind of like a math class. Teacher shows you the hard way to do it, then they teach you there's an easy way. But you got to take your lumps. You got to go through the hard way first. Okay. So we're going to have parent number 1 look like this. What would you call the genotype of this parent? Anybody want to take a shot? Yeah. You would say heterozygous, heterozygous, or you could just say a true heterozygote because they're heterozygous for both. If they were homozygous dominant, you could call it a true homozygous dominant. You could call it a true breeding tall green. There's a lot of different combinations. It could be dominant hetero, hetero dominant, hetero hetero, dominant dominant, recessive hetero, recessive dominant, just so many different combinations. I think there's 8 total. And that gets me to my next point. How many different types of gametes could this individual produce? And that's actually a question on your mastering. There's 2 ways you can figure this out. Tactic 1. And this is only for dihybrids. Do the FOIL method. First, outside, inside, last. So if you did first, that would be dominant T, dominant G, outside, dominant T, recessive G, inside, recessive T, dominant G, last, recessive T, recessive G. So how many different gametes can be made when you have this number of traits in 2 different forms? You can make 4. So the answer would be 4 different gametes. Now, tactic 2, which is applicable for everything. If you did a trihybrid, tetrahybrid, pentahybrid, hexahybrid, octahybrid, crazy number of combinations, you just do 2 to the x power. X equals the number of traits. How many different traits do we have in this example? We have height, we have color. So it's going to be 2 to the 2nd power. That's going to equal 4. That's how you figure it out. You have several of those on your homework. So just to show me you know what you're doing, what if I had a trait like this? Yeah, that's crazy. How many different traits are there? 1, 2, 3, 4, 5, 6, 7, 8. What would you do? If I said how many different types of sperm or egg could you make? 2 to the 8. It's like 256, I think. So, to make our lives a little easier, I'm going to go ahead and tell you that parent number 2 is the same. But guys, hear me now. Listen, watch this. I could do this. I could do this. I can do any combination. What I want to impress upon you all is, one, we're going to need practice with this, and two, every situation is different. In this situation, we're going to say that the other parent is also a true heterozygote. And so what I need you to do is I need you to make a dihybrid Punnett square. This is going to be 4 by 4. And you are going to put the different types of sperm on one side and the different types of eggs on the other. Okay. Here's what we're going to do. I'm going to give you about 3 minutes to put this together, but it's not that simple. You're not just going to put TTGG. Here's what you need to do as well. This is really important for Monday's class. I need you to tell me what percent are going to be tall and green, tall and yellow, short and green, and short and yellow. There are 16 total boxes, so it's going to be out of 16. You then need to tell me the percentage, or the frequency, point something, 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 something. So you all should have a calculator on your desk. This is what I'm asking you to do. This is the job that you have been tasked. I'll give you about 4 or 5 minutes to get this figured out. We don't have a whole lot of time, but this is necessary time, so don't waste it. I'm going to unplug my computer so you cannot just copy off my work. I'll put it back up in about 4 or 5 minutes, okay? I'll give you a solid 5 minutes. I think that's only fair. You got this, Mariah? You got the setup? All right. Say bye-bye. You have 2 minutes on my watch. All right. That's 5 minutes. Let's look at the Punnett Square first. So I'm going to identify all the ones that were tall and green with a green highlighter. For row 1, it should be all of them. For row 2, it should be box 1 and 3. For row 3, it should be box 1 and 2. And for row 4, it should be box 1. I think did I do something wrong? Nope, that's fine. Okay. So how many out of 16 are going to be tall and green? 9. What is 9 divided by 16? 0.5625 or 56.25%. Okay. I'm going to use a yellow highlighter to identify how many are going to be tall in yellow. It should be row 2, box 1, row 2, box 4, and row 4, box 2. That's 3 out of 16. What percent is that? 0.1875 or 18.75%. I'm going to use a blue highlighter to identify the ones that will be short in green. That should be row 3, box 3 and 4, and row 4, box 3. That's 3 out of 16. That should be 0.1875. Yes?
I'm so confused about the box row 1, column 4, combining the homozygous dominant TG and the one in the top part.
I messed that up. It should be heterozygous for height and heterozygous for color. Okay, that was my mistake. Good eye. And then the last one, short in yellow, should be in the bottom right corner, row 4, box 4. That one should be 1 out of 16, which is 0.0625 or 6.25%. So here are my results. Now let's say that you are taking the AP exam and it tells you that you have 50,000 pea plants. And it tells you if you go by the ratios and by the frequencies, how many are going to be tall and green? What would you do to figure that out? Anyone? Yeah. You multiply the total times the frequency of whatever you're trying to find. So in this case, if I want to know the frequency, how many tall and yellow, or excuse me, tall and green, I would do 50,000 times 0.5625. And let's see what that is. That would be 28,125. So this is how Gregor Mendel used math with biology. This is why. It's all about statistics. So that's the hard one. You want to know the shortcut? Let's do that. Let's say I gave you guys a cross like this. You would call this parent heterozygous height, homozygous recessive color. Or you could say heterozygous tall, true breeding yellow. For this one, you could say true breeding tall, true breeding yellow. So I want you to cross these two, and you're like, oh, I'm taking a 90-minute session on the AP exam. I don't have time to do a dihybrid cross. Here's what you do instead. You do two small Punnett squares. You make one of them for height. You make one of them for color. So take a look at the first box, or the first Punnett square. What percent are going to be tall? 0.5. What percent are going to be, actually no, it's going to be 100. Yeah, excuse me, 100%. And what percent are going to be yellow here? 100%. So this makes it pretty easy. I gave you an easy one. I didn't mean to do that. If I want to know what percent are going to be tall and yellow, here's what you would do. You just do the tall times the yellow. So you would do 1.0 times 1.0. That equals 1.0, 100%. I gave you an easy one. I didn't mean to do that. But you multiply the frequencies. So you just do a simple Punnett square. So I think it's only right that we do the exact one that I just did on the up above. So let's do that. Because you guys don't really learn anything from that. That's too easy. Let's try this. Let's just do what we did above. True heterozygous times true heterozygous. Rather than do a dihybrid cross, do two monohybrid crosses. Okay. Tall is 0.75. Short is 0.25. Green is 0.75. Yellow is 0.25. And so, if I want to know how many are going to be tall and green, what you do is you just multiply the frequencies. 0.75 times 0.75 equals 0.5625. Same as the dihybrid cross. If I did tall and yellow, 0.75 times 0.25, if you did the math, 0.1875. Short and green, 0.25 times 0.75 is 0.1875. And then if you are as short and yellow, 0.25 times 0.25 equals 0.0625. The exact same frequencies as what you did above on the hard Punnett square. This is a whole lot faster. So I would recommend you do that. Work smarter, not harder. Are there any questions with this, with dihybrid crosses? No? All right. So let's get to the last topic for today. We have about 8 minutes or 6 minutes, so we should be able to squeeze this in. Polygenic inheritance. This is where two or more sets of a trait are responsible for the expression of the trait. So an example is skin color, everyone. Skin color in humans. It's not just dominant recessive. There are 6 alleles, 3 sets of alleles that actually dictate your skin color. If you are completely dominant, that's the darkest skin color there is. That's very dark. What do you think the opposite of very dark would be? It's an A word. Albino. Albino is no pigment at all. Do you know why an albino's eyes are pink? Because you can see the blue, or not the blue, you can see the blood behind their eyes. There is no melanin, there's no color. Melanin is what gives you your hair, eye, and skin color. The more melanin you have, the darker you are. Also, for all you sun worshipers, do you know why you suntan? It's protection. What type of skin is more protected against the UV rays? Light skin or dark skin? Dark skin. So if you are lighter skin and you are exposed to UV radiation, your body is trying to protect itself from the UV rays by releasing melanin, which makes your skin darker. So it's actually protection. And if you genetically don't have that dark skin all the time, it will eventually go away because you shed your skin and your normal pigment will be coming back. All right, so you can have a whole lot of different combinations. You know, just a few examples just to kind of mess around. You could have this. You could have this. You know, there are, I think, 64 different combinations you can have here. So it just goes on and on and on. How many different gametes can you form from those sets of alleles? 8. That's right. 2 to the third is 8. You can have 8 different types of alleles. 8 different sperm and 8 different egg. And what's 8 times 8? 64. There's 64 different combinations for this. So to finish off today's class, I'm going to ask you, what would happen if I had a parent that looked like this, cross with a parent that looked like this? Which parent would you expect to be darker, the left or the right? Yeah, they have 3 dominant alleles. The one on the right has 2. So here's my question. What are the odds they have albino offspring? So we have 2 minutes. Let's get this in here. What would you recommend doing? You want to do a tri-hybrid cross with 64 boxes in the Punnett Square? No, you're crazy. What could you do instead?
3.
Do 3. And you only want to focus on the recessive trait. That's what is going to lead to albinism. So in the A's, what percentage will be homozygous recessive? That's 0.5. No, 0.5. How about the B's? 0.25. How about the C's? 0.25. So you would have to do 0.5 times 0.25 times 0.25, that will give you the likelihood they have an albino kid. That would be 0.03125%. Or not percent, but that would be 3.125%. So it's pretty slim. Now, what if I said they had 64 kids? This couple had 64 children. How many of those children will be albino? You do 0.03125 times 64. 2. You would expect 2 out of 64 to be albino. So this is how math can really play into genetics. We'll do more of this on Monday with chi-squared.